It takes 1 or 2 minutes to go live in the YouTube.
Yeah, sure.
Yes, sir. We are now live in the YouTube. So, good morning, one and all. Myself, Dr. Sunil Kumar, Associate Professor at Sri Vishnu College of Pharmacy. I invite you all for the 5-day FDP program in SBCP. So, here is a college introduction video. So, with this one, I would like to welcome Dr. Mohan Verma sir to give a welcome note to the participants.
I thank the speaker for accepting our invitation. Thank you very much, sir. And dear staff and students, today we are having Raju sir. He is from IST Hyderabad. He is highly efficient and eminent in the area of organic reactions. He will explain you his topic for the FDP, emerging techniques in the area of the drugs. And we are very happy to have you, sir. I wish you all the best to all the students and staff who are attending this FDP. And I'm extremely thankful to all the speakers who accepted our invitation. And again, I would like to actually thank Raju sir for accepting the invitation. The main objective of this FDP is to impart new ideas, innovative ideas, and creative ideas so that we can upgrade and update our information in the area of the R&D. Thank you, sir.
Yeah, thank you. Can I start, sir, or anything?
So, with
can I start?
For 1 second, sir. Your introduction will be given by Dr. Yasser, sir.
Okay.
1 second.
Sure.
Good morning to today's speaker, sir, Dr. B. Chandraraj garu, on the gathering of the today's FDP program. I am taking this opportunity as a privilege to introduce our esteemed today's speaker of FDP session, Dr. B. Chandraraj garu. Sir is from the Bhadrachalam, Khammam district, Telangana. He did the BSc in the Osmania University, SR and BG Anna Degree College. He did MSc in Organic Chemistry at Usmania University, Hyderabad. He did PhD under the in Usmania University and carried out the research work at CSIR, Indian Institute of Chemical Technology. He did the postdoctoral fellowship under the guidance of Professor H.C. Brown, Center for Boron Research, Department of Chemistry, Purdue University, USA. Presently, Sir working as a senior principal scientist and head of the Department of Natural Products and Medicinal Chemistry, CSIR, Indian Institute of Chemical Technology, Hyderabad. Sir specialization in the research of synthesis of natural products and heterocyclics, medicinal chemistry, and natural products based bioactive compounds, development of novel synthetic methodologies. Sir awarded 11 PhDs and guiding presently 3 PhD students. Sir guided PG students, either MSc as well as MPharm students, about 48 completed. Sir also have the technology development and transferred about on two categories. One is the one pesticide developed by the Sir, that is glyphosate, and one pyrethroid molecule developed by the Sir, that is the S-phenvalerate. Sir has 104 research papers in national and international reputed journals. 23 patents he has in his credit. Sir involved in the invited lecturers about 30, and he attended lot of symposiums and conferences about his history. Sir is an editorial board member of the 3 national and international journals: Oriental Journal of Chemistry, International Journal of Chemistry and Applications, E-Journal of Chemistry. Sir also a peer reviewer of the 19 journals. Amongst the most important of 19, Journal of Medicinal Chemistry, ACS Omega, Organic and Biomolecular Chemistry, RQO, and Indian Journal of Chemistry, Section B, Combinatorial Chemistry and High Throughput Screening. Like that, several journals present administration responsible coordinator skill development CSR ISCT Hyderabad. He is a member FSSAI New Delhi. He is a member HRMS CSR ISCT. He is a member of ISCT Research Foundation CSR ISCT Hyderabad. Member Selection Committee Project Fellows for Assistant at CSR ISCT. Member Publications Patent Guidelines Committee CSR ISCT. Member ACSIR Course Curriculum Committee, CSR ISCT Hyderabad, internal examiner and external examiner for PhD Vivavos as well as the PG practicals, Usmania University, Kakatiya University, and Palmer University. Thank you for giving the opportunity to introduce for the today's speaker, Dr. B. Chandra's garu. Thank you very much, sir.
Yeah, shall I proceed, sir? Sunil Garu?
Yes, sir. The session is over to you.
Yeah. At the outset, I thank the introducer for the very nice introduction about me. And first of all, I thank the Vishnu College of Pharmacy, Bhimavaram, for giving an opportunity to interact with the faculty as well as the students on faculty development program. And today I would like to speak on the topics of the natural products and medicinal chemistry and the synthesis of heterocyclic compounds, how best we can prepare the natural products and heterocyclic compounds in a simplified manner. So, Dr. B.V. Raju, Padma Bhushan, is an eminent person and the founder for the Vishnu College, and we had an opportunity with the Vishnu College also. We have done the skill development program for the Bhimavaram College, and it is an opportunity to give a lecture on emerging technology in the field of the healthcare and pharmaceuticals. The Vishnu College is conducting the program, and I am very happy to interact with the faculty as well as the students. Thank you, sir, for giving opportunity. So please go to next slide, sir. Shall I, sir? Can I operate myself, or you have to operate?
Sir,
You will be.
if you want to turn the slide, tell me. I will turn the slide.
Yeah, please.
Shall I
Turn
go?
the slide, sir. Yeah.
Next one, sir.
Yeah, next one. Yeah, this is the CSR ICT where I am working as the senior principal scientist as well as the head department of the chemistry. So I would like to say a few words about the CSR ICT and its contribution to the society and nation. So my project director is Dr. D. Srinivas Reddy, Director CSR ICT. Sir, please next slide. The CSR system organism is like this. Our president is prime minister and vice president is always minister of the science and technology. We have the governing body members and the director general in the CSR headquarters in New Delhi and the director CSR ICT, Dr. D. Srinivas Reddy, like that. 37 laboratories, CSR laboratories are available throughout the country. And we have the research council as well as the management council to guide the director to run the systems. Next slide, sir. Yeah, actually, it is a brief introduction about the CSR ICT. CSR ICT is already completed 80 years in its era. In 1944, we started the CSIR institution, and it was coal research is started in 1944, and simultaneously in 1950 we started the research on ceramic. 1960 we started the research on the oil and fat surface coatings. Comes to 70 agrochemicals we worked on healthcare, and 1980 generic drugs, and 1990 we started the lipids and polymer area. 2000 semichemicals, and the environmental ecology we started in 2010, and we also started the functional materials in 2017, and presently we are working at 22-23 on pharmaceuticals as well as the agrochemicals in addition to the environmental healthcare. So this is the journey where it was started from the central laboratory to regional research laboratory. Now presently it is called CSR IICT, Indian Institute of Chemical Technology. So next slide, please. And these are the areas where CSR ICT is working, and we have total 160 scientists are working in CSR ICT, including technical staff, and research fellows are more than 500 students. And we are working in the area of the healthcare, chemicals, energy, and environment. We have the department like fluoro and agrochemicals, polymers and functional material, organic synthesis and process chemistry. Myself is Dr. Chinaraju, head department of the natural products and medicinal chemistry, where I am working in the department. And comes to catalysis and fine chemicals, scientists are working in that area. And comes to energy and environmental engineering, oils, lipids, science and technology, analytical and structural chemistry is most important to do research. Also, we have that department. And comes to the applied biology, where we are working on the chemistry, synthesis, and medicinal, where medicinal chemistry involves a lot of biology, so that we have the dedicated biology scientists in the applied biology. Then comes to chemical engineering and process technology are there, where we are working on the process technology like pesticides and as well as the medicinal chemistry and pharma. The chemical engineering play a major role, and we need to develop the technology, so that process technology is most important. That's the reason CSR ICT is having so many departments to perform and to deliver the goods to the nation and society. Thank you, sir. Next time. Next slide. Yeah, I would like to say about the skill development programs which we are conducting at CSR ICT, because I am the nodal officer for the skill development program. We are also conducting the skill development program for the Andhra Pradesh government, and we are also conducting the skill development program for the V.V. Raju College, Bhimavaram also. So that's the reason I would like to say about a few words about the skill process. We are conducted almost skill development programs, 9 programs we are conducting, and the total beneficiaries are 1,386, including Andhra Pradesh government. We had MOU, and we have the category for the skill development program. Like we are also conducting national, international programs, and capacity building to the students, training to the trainers, and industry personnel also coming, and some government officials are coming to take the skill development program in CSR ICT, and we have the basic cheminformatics and bioinformatics. This is the course we are conducting for the BV Raj College. Hello, somebody is. Hello, somebody is talking. Can you ask them to mute? Yeah, next we are conducting the advanced synthetic organic chemistry. It is a 6 weeks course, and the another program what we are conducting is animal cell culture technique. That is 6 weeks we are conducting, and another program advanced cell and molecular biology and analytical techniques in food and neutral chemistry. Another program we are conducting for the 2 weeks, and another program for the those who are completed the BTech and MTech, that is process plant drafting using AutoCAD. And a simple lab technician course also we are conducting, because those who are studied the 10th class and intermediate level, we need to give training to go there to settle in industry in the lower cadre as a chemist, a lab technician. This is very important course what we are conducting for the intermediate. And comes to PG students, we are coming from the northeastern. They are coming to the CSR ICT, and they are undergoing for the training for the 12 weeks for the chemistry particularly. And another program what we are conducting is water purification and analysis for the 4 weeks for the engineering students as well as the ITA students. And finally, we had a MOU with the Kiwani and Arazan company recently, and we successfully completed one program. And just 2 days back, we distributed the certificates. And this program we conducted for the students 4 weeks, 4 months, and those who are completed the MSc, so they are ready to go to the industry. So these are the skill development programs we are conducting at CSR ICT to the benefit of the students to enhance their skills. So next slide, sir. Next slide, please. Yes, sir. Now I would like to come to the main theme of the today's program. Yeah, we were talking about the drug development, drug discovery, natural products, and synthesis. Simply, I would like to start with the paracetamol. Everybody knows that paracetamol is a drug used everywhere in every hospital, every patient. So it is a simple compound. It starts from the benzene, and it is available byproduct as the petroleum industry. And benzene is converted to the phenol. Everybody know that. And phenol on nitration gives the paranitrophenol, and nitration reduction gives the amino, and acetylation gives the paracetamol. This is the simple chemistry where paracetamol is synthesized. But when you talk about this molecule, for some time the reduction process is difficult, and catalysis is difficult, and acetylation process also. Sometimes if you use acetyl chloride, that is cost effective. For example, if you take acetic anhydride, that is a banned chemical, then how do you do acetylation? So that is a challenge. Can you do with the acetic acid? Yes, can be done. Instead of the acetic anhydride, acetyl chloride, can we use acetic acid? Yes, that can be done, so that the process will be lower and the cost will be lower, and you can use some catalyst where the acetylation can be taken. So this is also one of the important reaction. You cannot say that it is a simple. You can, if you can develop a catalyst as well as the with using acetic acid, this can be done, this particular step, and its requirement is almost 12 metric ton for the year. So India will be able to export now the intermediate as well as the final product. Once upon a time, we used to get the raw material from the outside, but now we are able to do it everything in the India. So this is the drug enhancement and drug development. It is a simple compound, and I would like to go for the complex molecule in another slides. Thank you, sir. Next one. Yeah, then if you talk about the what is the drug and drug discovery, everybody know that it is a defined composition with a defined pharmacological effect, whether it is a cancer or tuberculosis or diabetic. Then it has to be approved by FDA. Then only we can use as a drug. Then what is the process of drug discovery and development? Yeah, drug discovery is a very complex process, no doubt, and several compounds you have to prepare or isolate to make them, to screen them, and to identify them. After identifying also, you have to go for the in vitro, in vivo studies, toxicology studies, another pharmacodynamic studies also. For that, you have to use huge money to develop a one drug at least. For that, everybody knows that a drug discovery, it takes almost 10 to 15 years, depend upon the molecule, manpower, or available sources, Then other parameters like just now I told you about the chemical engineering process or process development to achieve the process to transfer the industry, then come to the public. So almost it takes chemistry, including biology, including engineering, that almost takes 10 to 15 years. So this is the drug process. That's how drug discovery will be happening. So next. Yeah, drug discovery process involves the natural product extraction. For example, you want a small molecule or big molecule. The source is natural product. That means you can get the natural product from the fermentation broth. You can take the plant. You can extract the compound. To get the compound, you can extract the plants. And other sources, animal fluids, like for example, you can take the snake venom also to develop the drug. And comes to synthetic chemicals. For example, you want to synthesize the molecule or small molecule or natural product extract, you can synthesize in the laboratory by using the medicinal chemistry purpose. You can derivatize, you can synthesize, or you can make the combinatorial chemistry so that you can make the molecule. Comes to biology. For example, you want to take the isolation, but means some broths are there. You can isolate the natural product from that also. You can use the recombinant process. So recombinant means product made in the laboratory using a technology. So that way you can make the compounds so that you can make the drug discovery and drug. Next slide, sir. Yeah, we are talking about the drug discovery means not only one chemist or biology or analytical people will be involved. If you see this slide, there is a physicians are involved and molecular biology involved, pharmacology, psychology, statistics, toxicology, formulation also most important, immunology, enzymology, biophysicist, biochemist, chemical engineering, spectroscopy, analytical chemist, medicinal chemist, then process chemist, metabolic, then molecular model, you require synthetic, then crystallography, scientific programs you have to conduct, then chemical information specialist should be there, and you have to make a data of the biology. Then clinical research are most important. Then hardware, software, including computational biology. That means you want to make a drug discovery. These many people are involved to make it one drug. Okay, sir. Next. Then I would like to show some complex bioactive molecules. Everybody knows that anti-cancer is one of the cancer is one of the disease. For that, we have to make the cancer compounds or anti-cancer agents. That is, you can say that belanol is one of the important compound for the anti-cancer. Artemisinin, everybody know that artemisinin is one of the important drug for the anti-malaria. And discodermalide, one of the important drug for the anti-cancer. And abalactone, anti-cancer. Rameshwarite, this is the compound isolated in the CSR ICT by Dr. Venkateshwarlu. He put the rameshwarite and it is used as the anti-asthmatic compound, and methanolide also antibiotic. So these are the very simple complex molecule discovered for the anti-cancer, malaria, and as well as the anti-asthma and antibiotic. Next slide, sir. Next slide, please. Yeah, I would like to show some simple compounds. Looks like it is simple. It's not a simple. It is the cyclopentane ring is there, or pentanone ring is there. If you see that, these are very, looks like that similar compounds, similar structure is there. One is the olefin is there, another one ester is there, another one carboxylic acid, hydroxy group is there, another one is ketone with the hydroxy. And if you see that carbophos, it is a anti-xylotic properties displaying. When it comes to misoprostol, one having the keto functional group as well as the hydroxy having a olefin with an ester, and it is used for the medication for the abortion. And latanoprost is one of the important drug for the used anti-glaucoma, where our eyes people are suffering with the eye problems. So these are the looks like the similar molecule, but the then functional groups are different in some carbon, so that it is acting a different activity, and one is showing the anti-glaucoma, one is used for the abortion, then another one is for the anti-oxalic acid, oxalic property. Then if you see another natural product derived from the plants, one is warfarin. Everybody knows that it is an anticoagulant used to treat for the treat the and prevent and treat the blood clots, and as well as the dicumarol is also similar compound where it is used for the anticoagulant treatment for the thrombosis, and wherever blood clotting are happening, So it is a disorder that can be reduced, that can be minimized or reduced by using the warfarin as well as the dicomera. So these are the simple compounds and complex compounds isolated from the natural products, isolated from the plants, and it is used. Next, sir. Yeah, if you see this slide I took from the I took the slide from the Google, where it is showing the natural product, tree like natural product. You can see this heterocyclic and heterocyclic. That means a compound having the nitrogen function, nitrogen moiety, and the O heterocycles. That means oxygen containing the heterocycles, and carbocycles. That means totally carbons are there. So that you can make the carbocyclic and nitrogen containing heterocyclics and oxygen containing heterocyclic. There are several compounds are isolated, and all the compounds were shown in this particular chart. The next slide. Yeah, now comes to the topic. I would like to say that what are the bioactive compounds can be made from the simple chemicals, and for that I would like to now tell about the what is the retrosynthesis, how to make the compound, simple compound or complex compound for the natural product, as well as the synthetic compound, and some examples I would like to show where the process development of the heterocyclic compounds are so important to develop the drug to transfer to the industry. Then novel heterocyclic compounds, how can it can be made from simple chemicals. That's what I would like to focus my topic now. The next slide, please. Yeah, now look at up the synthesis. What is synthesis? For example, synthesis means you have to make the compound. So how many syntheses are there? That means you can say there are types of synthesis are available to make the molecule. One is linear synthesis. What is linear synthesis? A total molecule made through a series of linear transformations. That means you can continuously do the reactions, continuously do the steps so that you can get the molecule. Then what is a convergent synthesis? Convergent means individually prepared compounds are convergent, brought to a make to the target compound. That means you can make it a individual compound, make it a target compound. Then divergent synthesis. For example, you want to make some intermediate. You are in the process of making some drug intermediate or some heterocyclic compound, or some you prepared some lactone, for example. From that lactone, you want to make a different type of the compounds. That means diversity you can make it. So you can make the simple common intermediate. You can make number of compounds or series of the compounds. So that can be useful for the synthesis as well as the molecule. You can make for the drug candidate, and biological activity also you can test it for that. Next comes to partial synthesis. That means you started some molecule, you are unable to end it. But instead of completing the total synthesis, you can make the partial synthesis. You can make the intermediate. Intermediate can be tested for the biological activity. Mostly that intermediate compound also can be useful for the some biological activity, so that you can develop for the drug also. Then comes to biomimetic total synthesis. Presumably, nature will not using a process that is intrinsically difficult or impossible. Then you can make it. You believe it that one can effectively mimic the condition provided by the nature and conduct some reactions in the plasma, so that you can make the compound. So that way you can make the synthesis by using the linear, convergent, divergent, total synthesis, or partial synthesis, or biomimetic synthesis. This I would like to tell one by one with some examples. So next. Yeah, when talking about the linear synthesis, just now I took the coumarin, one of the coumarin compound. Just now I talked about the dicumaral as well as the warfarin. If you take one example, carbocyclic fused ring system, that having the antibacterial activity, anti-tumor activity, anti-HIV activity. So this can be conducted the reaction by using the simple reactions like Peckmann condensation, one of the name reaction, and another one is Michael addition reactions. That way, if you use these reactions, you can make these molecules. For example, you see the retrosynthesis. That's what I have given one molecule for the retrosynthesis, where just now I have told. Retro means you can make the final molecule by different by fragmenting the molecule. That means this molecule, for example, if you make by cyclization oxidation, the previous step is cross coupling. The previous step is the having the intermediate like chloroaldehyde. The another molecule or another intermediate is boronic acid. That way you can make this molecule. I have shown the retrosynthesis, how we can make this compound. So next slide. Yeah, this is the synthesis where I want to show. for the particular molecule, you can make the flavonone or flavone or carbocyclic compound with the ketone moiety, where you can do the reaction with the POCl3-DMF. This is the Wilsmeyer-Hack reaction, where this keto can be converted to chloro and aldehyde. And after that, you can do the Suzuki coupling reaction, where chloro is there with the anisyl boronic acid, and by using the palladium acetate, you can make the CC bond formation. Then followed by oxidation. That means aldehyde can be converted to an acid. Then acid can be converted to acid chloride. Then intramolecular cyclation by using the AlCl3, you will get the compound. So that way, this is the linear synthesis. Just now I told you what is the linear synthesis, what is the molecule you can make, how many transformations are there. So these can be shown, this type of reactions are shown in this particular slide. Next slide, sir. Yeah, this is another example I would like to show. That is a very simple reaction I have shown. This is now whatever the reaction I am showing is now for the stereoselective synthesis. That means you have the chiral molecule. It is a chiral molecule is there. This is a molecule used for the anti-cancer activity on colon cancer, breast cancer, as well as the lung cancer. And this is the molecule piperidinilyl dihydroxy decene, having the at fourth carbon fourth is isomer is S, and fifth one is R and E. That is, there is a double bond is there, that is E, and the fifth configuration is R, and the fourth carbon configuration is S. So this is the important molecule, and this can be achieved through Wittig elimination, stereoselective sharpless asymmetric hydroxylation, then epoxidation, trans-resoselective opening of the epoxy alcohol, Harner-Wordsworth-Immons sulfonation to make the double bond. So by using these protocols, you can make this particular molecule, and that too chiral molecule. Next one, sir. Yeah, this is the synthesis where you can start then ester compound having, for example, 1, 2, 3, 4, 5, 6, 7, 8. 7 carbon. You can take the 7 carbon olefinic ester. The ester can be converted to alcohol, and this olefin can be converted to epoxide, and this epoxide can be opened to get the hydroxy as well dihydroxy compound, and one of the hydroxy compound can make the benzylation, and followed by debenzylation, and followed by one of the hydroxy was protected with the TBS, followed by after protection of the TBS, you can deprotection, followed by esterification. Then ester is converted to acid. Acid was converted to reacted with the piperidine, so that you can get the compound. That means there is a 1, 2, 3, 4, 5, 6, 7, 8 transformations are there. That means in a single, when you start without making any fragment, continuously you can make the compound by chemical transformations using an ester, olefin, protection, deprotection, condensation reactions, so that you can make the dihydroxylation, so that you can make the molecule by using linear synthesis. This is one of the important molecule. Next one. Next one. Yeah. Yeah, another example I want to show. Now linear synthesis, I had talked about the linear synthesis. Now I would like to talk about the total synthesis. So what is the total synthesis? One example I have taken is one of the molecule, very important molecule is cherylene. This is a heterocyclic molecule, 1, 2, 3, 4 tetrahydroisophenylene, and optically active. This is the S isomer at C4 carbon, and this is isolated from the Crinum powelli. It is a Amaradaceae family. How it is prepared? So there are 2 fragments. You can make it to 2 fragments. One is the hydroxy compound. The another one is a compound having the benzene with hydroxy methoxy compound. For that, you can choose a simple compound like dimethyl phenol. The phenol can be converted to the allyl alcohol. Then allyl alcohol upon rearrangement. So Kleiser rearrangement, you can make the transformation from O-allyl to C-allyl. Then hydroxy was converted to the benzylation, and olefin is converted to acid. That means oxidation you can make it, and you can make the acid. The acid can be converted to ester. The ester can be converted to alcohol to brome. That means one of the fragment where this benzyl 2,6 dimethyl benzoyl oxy phenyl bromide, you can make it by starting from the phenol. Next slide. Yeah. Then another fragment where dimethoxy tosylamine, benzylamine is there. This can be prepared from the aldehyde, imine, then amine you will get, and tosylation. In 3 steps, you can make this particular compound. And under basic condition, whatever the intermediate we made in the previous slide, you can do with the basic reaction, condensation reaction, where this nucleophile can be generated here by using the sodium hydride. Simply you can eliminate the HBr so that you can make the N-tosyl compound. You can make the N-tosyl compound. Then removal of the benzyl, you can use the palladium carbon hydrogenation, where debenzylation is happening. You can get the phenol. Then after getting the phenol, if you can do oxidation by using the silver oxide and DCM, you can make the phenol method. This is called the phenol method, having a double bond O, having an olefin here. Then intramolecular cyclization with zinc chloride in presence of the DCM solvent, where you can get the cyclization. That means you can make the 2 fragment in this particular total synthesis. You can make the 2 fragment. One is the aniline fragment, benzylamine fragment. The another one is phenyl fragment. You can couple with these 2 upon oxidation, followed by first you have to do the hydrogenation, followed by oxidation, then ring cyclization. You can get the synthesis. That means total synthesis you can make the molecule by making 1 fragment, the another fragment. By taking 2 fragments, you can make the molecule. Next one, sir. Yeah, another example what I want to show, total synthesis of the marmosine. 2 examples I have taken now. You know, just now I have shown about the coumarin, 4-hydroxycoumarin. Coumarin is so important. So you can take the 1, 2, 3, 4, 5, 6, 7 hydroxycoumarin. You can make the allylation so that O-allyl or prenyl it will be formed. The prenyl under Lindell cut selective reduction, you can make the triple bond to double bond. Then under 200 degrees Claise environment, you will get the 2 isomers. One is the linear, one is the adjacent to the hydroxy. You can make the prenyl compound linear as well as the another isomer. So you can make this compound by using the MCPB oxidation. You can make the marmosine furan compound, where it is cyclized here in this compound. And if you can make another isomer, you can make the columbiridine. That means you can make the 2 compounds, marmosine as well as the columbiridine. So same oxidation by using the MCPBA, 2 intermediates, you can make the 2 compounds. One you can make the marmosine and another one you can make the columbiridine. But both are most important compounds. If you test it, both are biologically active compounds. Next slide, sir. The another one is, just now I talked about the biomimetic total synthesis. Now we have seen the total synthesis. Then what is the biomimetic total synthesis? Then one ester compound, just now I told about the total synthesis. Same ester compound can be taken having a olefin moiety. This olefin can be converted to the tribromo derivative. This tribromo derivative can be eliminated to the HBr elimination. You can get the olefin bromo compound. The acetyloxy compound, you can remove the acetyl to the hydroxy. Hydroxy can be converted to keto. Then keto, after conversion the keto, this bromo can be converted to dimethoxy compound. Then hydroxy compound can be made. Then intramolecular cyclization by using the immobilized Baker yeast as well as the sodium borohydride reduction, you can make the lipoic acid. That means in the laboratory, final step whatever it is there, up to this you can make it. But final step whatever the immobilized Baker yeast is there, under pH, if you maintain the pH 4.5 to 5, and by using the phosphoric acid and acetone solvent, afterwards sodium borohydride reduction, the ester can be converted to acid. You can make this lipoic acid. As I just now I told about the biomimetic, that means you can make the synthesis and the final molecule you can use the immobilized Baker yeast. In one step you can make the chiral compound. Next step, sir. Then another example what I want to show biomimetic total synthesis is, just now I have shown that a moiety can be made. This moiety can be made by simple phenol, dimethyl 2,6-dimethyl phenol. You can make the carboxylic compound. where I have shown heterocyclic compound previously. Now it is the carbocyclic compound. Similarly, you can make the instead of the silver oxide and instead of the silver oxide, if you can make it to the mushroom tyrosinase enzyme, you can make the same quinone method. Where I prepared the quinone method by using the zinc chloride and DCM there. Here I use silver oxide and zinc chloride where I used the intramolecular ring cyclization. Whereas here I used the mushroom tyrosinase with pH 6.2, 6.8 and with 6.8 pH in 50% methanol, estronitrile. So you can make the intramolecular cyclization to get the carbocyclic. That means you can make this particular compound in the laboratory. The final transformation to make the chiral compound, you can use the mushroom tyrosinase with, by maintaining the pH in presence of the acetonitrol solvent, you can make the chiral compound. So this is the biomimetic total synthesis to make the tetrahydronaphthalene derivative. That is this is the carbocyclic, that is the heterocyclic compound. Next, sir. Yeah, after all making the, just now I told about the linear synthesis, total synthesis, partial synthesis and biomimetic synthesis. Then how to make the heterocyclic compounds with the simple available chemicals in the laboratory. That means any faculty, how you can make the simple chemicals by using the simple chemicals. Some heterocyclic compounds are useful compounds. And the students also can plan their research with the available chemicals in the laboratory, how best I can make the compounds with the simple chemicals. For example, if you see this slide, this is a simple beta keto ester. Everywhere it is available in all the laboratory, not only in big laboratories, in small laboratories also, the beta keto esters are commercially available. That means it is a versatile and effective reagents. And there is a electrophilic center is there is a nucleophilic center. And it is a composed of the two electrophilic carbonyls are there. You can see that CH3 C double bond O, one ester group C double bond O is there, and two nucleophilic carbons are there. That means this C double bond O and this C double bond O. That means it is a very simple compound, but it is very fantastic molecule. I can say that. That R can be methyl, phenyl, CH2Cl, CCl3, and CF3. These are the available beta keto esters with the strong electronic withdrawing group, as well as with phenyl. Then comes to, if you see that just now I told about the Peckmann condensation. What is the Peckmann condensation? You can take the resarsenal. It is a benchtop chemical, and you can use with the ethyl ester in presence of the sulfuric acid or PTSA. So you can make the simple coumarin compound. That means R can be methyl, phenyl, trifluoro, CH2Cl. That means depend upon the beta keto esters, whatever you take, you will add the compound. This is a simple Peckmann condensation. Then comes to Navigal condensation. It is very 1940, 1960 reactions, I can say that. Now, what is the Navigal condensation? It is a simple salicylaldehyde, benchtop chemical. Same you can take the ethyl ester, and if you use the piperidine or DCM, you can make the 3-substituted coumarin. This is the simple Navigal condensation. That means you can make the methyl compound, C double bond acetyl compound, phenyl compound, or if you use malonic acid or malonic ester, you can make the ester compound as well as the acid compound also. That means 3-substituted coumarin you can make. That means 4-substituted coumarin you can make the Peckmann condensation, and 3-substituted compound you can make the Navigal condensation. Next. Yeah, this slide shows that what is the novelty of the compound here, what we can make it novelty of the compound. Just now I told that it is a simple salicylaldehyde. Yes. Instead of the ethyl acetate or methyl acetate, you can take the chloroethyl acetate. That means instead of the CH3, you are introducing the CH2Cl. That means electron withdrawing group you are introduced. That means under Navigal condensation reaction, that means it is a simple piperidine or base catalyzed reaction. In DCM at room temperature, you are supposed to get this compound. That means C double bond O CH3. In this case, you will get the C double bond O CH2Cl. But this compound we could not get. What could be the reason? What could be the reason? There is a reason. Then we expected this compound, we didn't get it. And this compound, whatever the down I have shown, this we got the compound. That means 2 to 3 trisubstituted compound we got, and that means the reaction went in a different way. The reaction went in a different way. That means here the piperidine is the one of the base utilized, and you can use the pyridine, you can use the DMAP, DAPCO, imidazole, triethylamine, isopropylethylamine, sodium methoxide in methanol or ethanol, potassium carbonate, N-disopropylethylamine. I mean, another base you can use DAPCO also. That means these many bases you can use to get the compound, but none of the base has given the required compound. No, whatever the seed of SL compound, we could not get it. We got this compound only. I will tell the reason why we got this particular compound. So we use the solvents like acetonitrile, methanol, ethanol, chloroform, DM, toluene, DCM. In any solvent, in any piperidine, we could not get the compound, that compound, SL compound. We got only 2 to 3 trisubstituted compound. What could be the reason? Next slide, sir. Yeah, this reaction actually, whatever the anion generated here by using the piperidine, it should attack on the ester compound. But instead that, whatever the C double bond O CH2Cl is there, it approached, a nucleophile is approached to this carbonyl rather than the ester carbonyl. Why means this is the chemoselective reaction. It is the chemoselective. That means selectively the nucleophile is approaching the electrophile, that is C double bond O CH2Cl, instead of the C double bond O ester. So that is the reason you got the particular compound instead of the other compound. It is a nominal derivative only, no doubt at all. We can say that it is a nominal conjugation, but instead of getting the acetyl compound C double bond O CH2Cl, we got the 2 to 3 trisubstituted coumarin compound. We got it. That means it is a chemoselective compound. That means a simple salicylate, when you change the beta ketoester, you got the different compound instead of getting the whatever the compounds are reported in the literature. Sir, next. Yeah. So when you do this reaction, we got this is the 2H chromine. We can say this is a 2H chromine compound. Then 2H chromine compound, how best we can convert it to 4H compound? Because there are no reports to convert 2H chromines to 4H chromines. Then we decided how best we can convert this, because it is a simple water elimination, you will get the compound. Okay. Then what are the reagents we can use? We can use the Lewis acid like BF338, FeCl3, AlCl3, copper iodide, copper chloride, even iodine and PTS also we used. But none of the reactions has given the good yield except BF338. In DCM, we got the 4H chromine. So that 2H chromine can be converted to 4H chromine. How best we can convert? It is a simple reaction. BF338 is commercially available. You can make 2H chromine to 4H chromine. So why should we convert the 2H chromines to 4H chromine? Means yes, I have the answer. Here there is a CH2Cl, there is a functional group Cl is there, you can replace it. There is a ester group, you can play it. That means 2 functional groups are available in the same moiety. That means you can do the chemical conversion so that you can get the another compounds or different compounds. For that reason, 2H chromine is converted to the 4H chromine. Next one. Yeah, this is the mechanism where I have shown how BF3 etherate is helping to convert 2H chromine to 4H chromine. Next, sir. That means it's nothing but it is forming the oxonium ion, then eliminating the water, approaching hydride from the C4 rather than the C2. Then water is eliminated and it was given the compound. Next one, sir. Next slide, please. Yeah. So whatever the 4H chromine is, what we got from the salicylaldehyde, then how best I can convert it to another heterocycle? Yeah, there is the answer, or this slide is showing like this. For example, just now I told there is a moiety CH2Cl and there is an ester is there. How best I can convert it to this to another molecule? So if you condense with the benzylamine, it is a benchtop chemical. Benzylamine is everywhere, it is available. If you take a benzylamine and you take this 4H chromine and you condense in presence of the ethanol, just without any reagent, any catalyst, you can simply reflex in ethanol, you can get the chrominopyrrolones. That means you got the fused compound. That means you can make another heterocycle. So this is an important transformation reaction with the simple chemical. Next, sir. Then comes to, instead of the benzylamine, if you take a propargylamine, That means you can make the propargyl. Then if you do Kilk reaction, you can make the triazole. So another cycle, another heterocycle you can make with the triazolmide. Triazole compound you can make it with the azide compound. Next, sir. Next, sir. Next. Yeah. Similarly, 2H chromin was converted to the pro-H chromin. Then 2H chromin, how best I converted to another heterocycle. That means if you take a 2H chromin, if you react with the simple salicylic, whatever the salicylic just now I told is a benchtop chemical, in presence of the BF3 we got the spiro compound. Very difficult to get the spiro compound. Very difficult to get the spiro compound. But with this reagent like BF3, same reagent, we use it for the transformation in presence of the instead of the AlCl3, FeCl3 or TFA or PTSA, BF3 was given the best yield at -78. We got the spiro compound. Very important heterocycle compound. Next, sir. Yeah, this is the substrate scope where we have shown that the 2H chromin converted to these spiro compounds. Next slide. Next, sir. Yeah, this is the mechanism where I have shown how the transformation is happening with the BF3 thread. That means initially water is eliminated. The another molecule of the salicylaldehyde again it is condensing and giving the spiro compound. Next, sir. Yeah, just now I have shown that 2H chromin to 4H chromin and 2H chromin to spiro compound. Then again 2H chromin, how best I can convert it to other heterocycle, that is benzoxapine. This is the benzoxapine, that is a 7-membered ring. That means when you use a Wittig reagent, you know Wittig reagent, everybody knows the importance of the Wittig reaction. If you use Wittig reaction, Wittig condensation or Wittig homologation reaction, if you do reaction with the homologation with the 2H chromin, we got the ring opening and ring closing reaction to give the benzoxapine. That too with the cis selectivity, not a trans selectivity. Selectively we made the cis compound rather than the trans compound. So we used the N,N-diisopropylethylamine. This is the best base we utilized to get the compound benzoxapine. Next. Yeah, that's how it went. This is how the reaction went. The 2H chromin was converted to the benzoxapinone. That means ketone is formed. With this, first initially the ketone is formed. Then this ketone underwent the Wittig homologation. We got the compound. This is what I showed the transformation. This is the mechanism. This is how the reaction is going. Next, sir. These are the examples shown how to make the compounds. Different electron donating and withdrawing groups you can introduce on the phenyl ring, as well as the Suzuki coupling reaction also. You can make the pyran and furan ring also, or other heterocycles also can make it. Next, sir. Yeah, just now I told about the benzoxapine, how best we can make the benzoxapine with the simple salicylaldehyde. That means salicylaldehyde is reacted with the chloro ethyl acetoester, you got the 2H chromin. The 2H chromin can be converted to benzoxapine. From benzoxapine, how best we can make the other heterocycles. Next, sir. This is a very simple reaction. Benzoxapine, you have the ketone, C double bond O, the C1, C2, C3, there is a ketone, C4, there is an ester. That means you have the ketone as well as ester. So you can use the hydroxylamine hydrochloride in presence of the ethanol and acetic acid. You can make the a important compound, that is isoxazolone. That means isoxazolones are very important compounds in the drug discovery. It has shown the anti-cancer activity as well as the antibacterial, antifungal activity. With the benzoxapine having a keto and ester, if you condense with the hydroxylamine, you can make the isoxazolone compound. Next, sir. If you instead of the hydroxylamine, if you make the phenylhydrazine hydrochloride, it is also a benchtop chemical. A benchtop chemical. It is everywhere. It is available. Everywhere it is available. You can take the benzoxapine and phenylhydrazine hydrochloride in presence of the ethanol, 1 drop of the acetic acid or 1 ml of the acetic acid. Under reflux condition, you can make the pyrazolone. where earlier compound you got the isoxazolone, here you can prepare the pyrazolone. So that means a different compound you are making. Isoxazolone as well as the pyrazolone, you can make the compound just by simple condensation reaction with the hydroxylamine and phenylhydrazine hydrochloride. Next compound. Next slide, sir. Yeah. How you can make other heterocycle by taking the benzoxapine? Simple reaction. Now this is also a very simple reaction. Orthophenylene diamine. Orthophenylene diamine is a very benchtop chemical. It will be available in anywhere. Whatever the benzoxapine is there, if you treat with the phenyl 1,2-diamine in presence of the Lewis acid, you can use the bismuth triplet. You got the benzophenazine. It is a benzophenazine, you got the compound. That means the ring is opened. The ring is opened. 7-membered ring is opened, and again it was cyclized, given the 6-membered ring, that too having the dinitrogen. That means it is a very good reaction to give a novel heterocycle compound. That means a 7-membered ring can be converted to a 6-membered another heterocycle compound by using the bismuth triplet, and another reagents whatever we used, the lanthanum triplet, acetic acid, sulfuric acid also utilized, but these compounds, these reagents could not give the better yield except acetic acid and bismuth triplet. Bismuth triplet got the 84% and acetic acid given the 72%. That means we found that bismuth triplet is the best catalyst to give this transformation reaction. Next, sir. Yeah, these are the previous reports. Very complicated reactions are available in the literature, but this is one of the simple reaction we converted to benzoxapines to the benzophenazine compound. And how the reaction is going? Anybody can, sir, how the reaction is going? Anybody can ask us. The reaction is going like this. That means benzoxapine is converted to a naphthol. Ester having the dinaphthol. And this OH is there. When you condense with the NH2 1,2-diamine, a simple water is 2 molecules of the water is eliminating, giving this compound. That means identifying the intermediate is most important. So we isolated the intermediate compound and we characterized by NMR. This is the NMR where the compound is characterized by NMR, and we proved that this is the intermediate is forming, and again isolated the compound. This isolated compound we condensed with the 1,2-diamine, we got the compound. That means it is proved. The intermediate is proved. How the reaction is going also it is proved. Next. Next, sir. Next slide, please. Sir, next slide. Next slide, sir. These are the phenazine derivatives. Next slide, sir. Next. Just now I showed about the reaction 1,2-diamine. That means you got the phenazine. If you take aminophenol, instead of the NH2, if you take NH2OH, aminophenol, the reaction given the phenazine under similar condition, if you take aminophenol, phenoxyzine is given the compound. And if you take the another compound, if you take instead of the cyclic 1,2-diamine, ethylenamine or propylenediamine, you will get the phenoxyline. That means if you take the diamine, you got the benzophenazine. If you get the aminophenol, you got the phenoxyzine. If you take the aliphatic amines, the 1,2-diamine or 1,3-diamine, you got the quinoxyline. That means the benzoxapine is opening and giving 3 type of heterocycles. One is phenoxyzines, benzophenazines, and quinoxylanes. So this is very simple transformation reaction and giving a good heterocyclic compounds. Next. So next slide. This is how the reaction is going. How BF3 iterate is how bismuth triplet is helping the reaction. How the compound is forming. That's what I shown this one, and also I shown the mechanism also. Next slide. Next slide, please. Next slide, please. Next one. No, next. Previous slide, sir. Yeah. Same benzoxapine, we are converted to benzophenazine, phenoxyzine, and quinoxyline. Instead of the 1,2-diamine, if I use a benzylamine, it is also benchtop chemical available in everywhere. In presence of the copper acetate, instead of the quinoxaline, instead of the benzophenazine, instead of the phenoxazine, we got the entirely different compound, naphthoxazole. We got the naphthoxazole, like 5-membered, having the nitrogen and oxygen. It's a very, very fantastic reaction to get the naphthoxazole. That means a different compound we got it here. A different compound we got it here. Next. Next. Next, please. Next, please. Next, please. Yeah, these are the derivatives we made it. Next. Next. One second. Instead of the benzylamine, if you make the aniline, it is also benchtop chemical available everywhere. Instead of the aniline, instead of the benzylamine, if you take aniline, you got the, instead of the 5-membered, you got the 6-membered. That means phenoxazine we got. There naphthoxazine we got. Here we got the phenoxazine. That means a novel transformation is happening by just changing the benzylamine to the aniline. Next, sir. Why you are making this compound? Anybody can ask me, sir, what is the reason you people are making this compound? That means benzophenoxazines are very important compound for the fluorescent, to make the fluorescent dye, for the labeling of the biomolecules, where you can see that meldola blue, one of the important compound, and phenoxazine, another important compound, where that is shown or exhibited the lambda maximum at 540 nanometers and emission is 568 nanometers. And if you see that Nile red, Nile blue, yeah, another important compound, that is the phenoxazine compounds, where this lambda maximum is 554 and the lambda max emission is 638. These are the compounds were used day to day in the labeling of the molecules, used for the redox sensors, detection of the materials, and it is used in the textile industry, paper industry, paint industry, and mainly to make the pigment also. So these are the very important compounds involving in the DNA, where the dye mediate is there, electron transport, where the patients are going, they will inject, the doctors inject the dye, and they will give the, they will assess and they will take the picture, image picture, they can set the where the blocks are there. So that way, these are the fluorescent dyes can be used in the medical systems as well as the other fluorescent dyes. Okay, sir. Next. This is the graph where I have shown the photophysical properties of the compounds where we have shown. These are the two important compounds shown very important. Lambda maximum, wherever the Nile red blue is there, more than Nile blue, Nile red, it is shown. That means where that 538, 534 is there. These are the two compounds having the nitrofunctional group, and the dimethyl functional group. It's shown 627 and 603. So that means whatever the compound was prepared here, that can be used for the fluorescent dyes and in medical use. Next, sir. Next. Yeah. Earlier slide we have shown that when you take this alkylaldehyde, chloroethyl we didn't get the compound. Then trichloroethyl if you use it, we are supposed to get these two compounds. This also we didn't get. Then what is happening? Why it is not coming? For example, if you make it a salsalaldehyde to another derivative, that means tosyl compound or hydrazone compound, if you make it, can any reactions will be happening with this reagent? That's what I would like to show. So next slide, please. Yeah, the simple salsalaldehyde is reacted with the trichloro, and we got the 3 ester compound, carbethoxy compound we got. Actually, we are supposed to get this 2 to 3 trisubstituted compound, or as per the nominal condensation, we are supposed to get the 3 substituted. Trichloroacetyl compound we are supposed to get. But these two compounds we did not get. We got the compound. Interestingly, we got the important chemical transformation where 3-carbethoxy ester compound we got it. How we got it, I will show in the next slide. It is also a piperidine simple non-analytical condensation reaction. Instead of the trichloro, if you use trifluoro in the next reaction, for example, salsalaldehyde, trifluoroethyl in presence of the piperidine, we got the again a trisubstituted compound. Instead of the carbethoxy compound, we got the trisubstituted. Because why it is happening is due to strong electronic withdrawing nature of the trifluoro. Due to strong electron withdrawing nature of the trifluoro, we could not get the carboxy compound. We got the 2,2,3 substituted compound. If you use trichloro, we got the carboxy compound. There, next slide, I will show you how the reaction is happening. Sir, next. Next slide. Next, please. I will show the reaction, how it is going. Next slide. Yeah, this is what it is going. This is how it is going. See, simple non-alkyl condensation, it is approaching. Earlier slide also I showed, instead of the approaching the nucleophile to the carboxy group, it approached to the carbonyl C double bond O CH2Cl. There, here C double bond O CCl3. That means this here the chloroform is eliminating. The interesting is here the chloroform is eliminated where you got the compound. There it is difficult to get the eliminate the CF3OH. CHCl3 elimination is easy instead of the CHF3. CHF3 is very difficult to eliminate. That is the reason where CF3 compound you could not get, where if you use CF3 you got the 2,2,3 tri-substituted compound. If you take the trichloro, you got the carboxy compound. Here the eliminated species is chloroform. Next, that's the reason this reaction is happened. So this is one of the important compound. The ester is the coumarin ester is prepared. Yeah, this is the compound where I prepared. Just now I told bioactive compound. What is a bioactive compound? The compounds what we made, the coumarin compounds, chemical transformation reactions. It has shown the DPPH activity, alpha glucose inhibitor activity. That means anti-diabetic activity. It has been shown the coumarin compounds, whatever we prepared earlier. Next slide, sir. Next slide. Another important compounds what we have shown is this is the 2H chromin, this is the benzoxpine, benzoxpino-isoxazolones, where we got the phenyl hydrogen hydrochloride, where we got the pyrazolone compound, hydroxylamine, we got the isoxazolone. So these are the 4 important compounds we prepared. Next. These are the benzophenazines, phenoxazines, and quinoxazines were prepared. These are the bioactive compounds where it is used in the photo labeling agents. Next. Exactly, now I have shown that what is the bioactive compound, what are the natural product, how natural product can be synthesized, how you can make the linear synthesis, total synthesis, biomimetic, and how a simple compound, a simple intermediate can be converted to a novel heterocyclic compounds. For example, you want to transfer a process chemistry of the any molecule, how you can do it. That means this is the important imidacloprid molecule, a pyridine molecule. It has a 2 fragment. One is the pyridine moiety, and another one is the imidazole moiety. If you combine these 2, you will get the molecule. How you can make the nitroguanidine first intermediate? You can take the guanidine nitrate. If you treat with the sulfuric acid, nitroguanidine, you will get it. If you condense with the ethylene diamine, you will get the one of the fragment. Next. Next, sir. Next. Previous slide. Yeah, this is another intermediate. What I want to show you is the pyridine intermediate. How you can make it? This is the picolinate. It is commercially available. If you do oxidation, an oxide will be coming. If you treat with the POCl3, you will get the 2 isomers, 2 chloro as well as the 5 chloro. Then another intermediate, how best you can make only single isomer. Where you got the 2 isomers, it is difficult to make the process chemistry. It is not a cost effective. If you take the 2 intermediate, it is not a cost effective. You should make only 1 intermediate, then it is a cost effective. Then how can you make the 1 intermediate? Yes, you can change the other route. You can take the benzylamine. If you treat with the propanaldehyde in presence of the KOH, you can make the imine. And if you do acetic anhydride treatment, you will get the N-acetyl compound. If you do the Ilsmach reaction, POCl3, DMF, you can get the chloro intermediate. This chloro, CH3Cl2 photochlorination, you can make selectively CH3 into CH2Cl. That means you can make a selective route from the benzylamine. You can avoid the another isomer by using this particular route. That way you can make the process. That means process means Ultimately, you have to make the commercially available with the commercially available chemicals with the cost effective, so that only industrially available material. Otherwise, it is very difficult to make the industry process. Next. And no industry will come if you make only commercial process. Industry will come, they will take it and they will give it to the society. Then comes to finally, I want to make a another 5 or 6 slides. I want to conclude. Now do new dimensions for the cancer research target therapy. That means now we have seen the molecules like the natural products and whatever the molecules made for the industry, and so many molecules are available for the cancer, and still people are suffering with the cancer. Then what new dimensions are coming in the cancer therapy and what researchers are doing? That's what I want to show in another 5 slides. Then I would like to conclude. This is the target drug therapy. What is the targeted therapy? There is a two process are there. One is antibody drug conjugate, the another one is proteolytic target chimera. What is the targeted drug therapy? You can make the targeted drug therapy by using the bioconjugation. You can use with the linker. That means you can make the one linker and do with the bioconjugation. You can make the targeted drug therapy. Bioconjugation means the attachment of the one molecule to another. That means you can make a covalent bond, which one is of biological origin. That means you can take the one biological origin and you can make the linker, so that you can make the antibody drug conjugate. Why we are doing this one? It enhances the bioefficacy of the final molecule, and the properties of the final molecule can be modified. So you can take the PEG, that means polyethylene glycol is a solvent that increases the water solubility. That means a monoclonal antibody, that is bioconjugation, that targets a specific cancer antigen. So you should target the cancer antigen while not harming the healthy cells. That means whenever you are giving any treatment, you should not harm the normal or healthy cells. If it is harming the healthy cells, you cannot use that as a drug, a cancer drug. That means you can make selectively that target the only specific cancer antigen. Then heterobifunctional small molecule compound means which consists of a ligand. You can use a ligand for the target protein, a linker and ligand to recruit the E3 ligase. Next slide. Yeah, this in this slide I have shown this. This is the antibody, and this is the target drug. Whatever we made it. For example, you made one target drug. This is the target drug, and in between there is a linker. That means if you see the next picture, there is a amino acid links are there. You know what is the amino acids, right? Aspartame, tyrosine. There is a previous slide, please. Yeah, there is a tyrosine. Hydroxyl groups are available. Carboxylic acid is available. Amine is available. Thiol is available. That means you have the choice of the groups. That means amines to take it to the reaction. Which side you want to take? Amine, acid, hydroxy group, and thiol side. Sir, next slide. Sir, next slide, please. Yeah. Bioconjugation, that refers the, just now I told, one molecule to another covalent, and usually between the two molecules or three molecules, tracking the cellular. You can, by what is the advantage is you can tracking the cellular event, revealing the enzyme function, how the enzyme is functioning, and determining the protein distribution, how it is biologically it is distributing, and imaging specific biomarker, you can say that, and delivering the drug to the target cell only. So whatever the drug you are made, that should be delivered to the target cell only. That means you should not harm the healthy cells. This is the figure where I have shown that conjugating agent. This is the linker, conjugating agent, where that bioconjugation is happening. Next slide, sir. Then if you do this, there is some drawbacks are there. What are the drawbacks? Means renders to the hydrophobicity to drug linker moiety, not stable in blood circulation. It should be stable, but sometimes it is not stable in the blood circulation. Not only that, it requires the selective and simple chemistry. That means if you choose a molecule, a bigger molecule or any complex molecule, sometimes it may not work. It may work, it may not work at physiological condition. So these are the simple drawbacks are there. Then next what? Sir, next slide, please. That means these compounds can be made into click reaction or Michael-Condense reaction or Beckmann-Condense reaction or Navigal-Condense reaction or any Robinson reaction. Any chemistry can use to make the molecule. Next, antibody drug conjugate ADC. What is ADC? It's a non-cleavable that remains the conjugate until the degradation of the MBA internalization take place within the liposome and endosome. Once the target cell it reaches, release the drug depending upon the physiological environment. Next, sir. Next. Yeah, these are the linkers you can use. What are the linkers you can use? There is a linker for the disulfide. There is a linker for the dipeptide. There is a linker for the glucuronide. And ether you can use, thioether you can use, and you can use the simple hydrazone also. Now these are the compounds are making the Pfizer and Seattle Genetics and Roche company. They are all making these things, sir. These are the linkers they are producing. Next. And you can also make the linker. Not only these linkers, you can also make the linker. Next, sir. And PROTAC. What is the PROTAC? Proteolysis targeting chimera. These are the three scientists got the Nobel Prize in 2004, and they discovered the ubiquitin mediated the protein degradation. That means you can avoid the infection, because whenever you give any drug that kills the normal, that damages the normal cells or healthy cells, that means you should avoid the infective disease. But whatever the drug you use it should effectively target the cell, and it should cure the diseases. But if it is damaging, then there is a problem. If it is giving side effects, then there is a problem. For that, they prepared they invented the proteolysis targeting chimera. What is the proteolysis targeting chimera means? It will not do not harm the tissue. It is a DNA based, and recombinant means products made in the laboratory using the technology. And another advantage is avoid the infectious diseases. So that's what they want to tell by doing the linker to ligand as well as the recognize the moiety. Next. That means a PROTAC, proteolysis targeting chimera. That means E3 ligase recognition moiety, it will recognize, and there is a ligand and there is a linker. That means there earlier two processes are there, here three processes are there, but that means there is a linker between. The process of rapidly removing the unwanted or damaged protein. Whatever the unwanted cells are there or damaged proteins are there, that will be removed. That means PROTAC degrades the target protein through the ubiquitin proteasome system. It binds the target protein and E3 ligase to form a ternary complex, making the target protein with the label of the ubiquitination. That means that avoid the infectious disease. Next, sir. Next, sir. Next slide, please. Yeah, these are the examples are shown here. This is a linker where I shown the red that nitrogen heterocycle is there. This is the linker. This is the linker. That means presently in India, in abroad they are making. Abroad they are making, Germany, Japan, and other countries are making. But presently in our India system, that Biocon is making this type of research now. Now other companies also slowly coming. That means this is the specific drug specific curity of curing the cancer therapy. This is now currently people are doing for the research, and anybody can take up the research on this angle, and they can make the drugs for the cancer therapy. Next slide, please. Next slide, please. Yeah, with this I would like to conclude my talk. Now finally the conclusion is whatever the B.V. Raju garu, Padma Bhushan B.V. Raju garu, Sri Vishnu Educational Society, today they have conducted the webinar for the to give the seminar on the to benefit the faculty as well as the students. And now I talked about the natural products, isolation, earlier drugs, present drugs, how the synthesis is making the linear as well as the biomimetic synthesis, total synthesis, how the chemical transformation can be done, whatever the chemicals are used, whatever the chemicals are available in your college or in your university or in your institution, a simple compound can be converted to a useful heterocyclic compounds of the biologically important. Now, what are the cancer therapies are available? Now, what are the recent trends is happening in the cancer therapy? What is that happening in the India and abroad? What cancer therapy you can make it, and you can make the new drug development for the cancer therapy. That's what I cover, sir. With this, I would like to thank the Sri Vishnu College for giving an opportunity to deliver a talk on this particular topic to benefit the students as well as the faculty. I thank once again B. Raju Garu Society, Sri Vishnu Education Society, and I thank the Vice Chancellor of the university, and I thank the Nataraj sir and Sunil sir. Thank you. Thank you very much.
Thank you very much, sir, for enlightening all the participants and us about the importance of medicinal chemistry and usage of the simple chemicals to produce novel heterocyclic compounds. And you basically told us how to convert a sim or utilize normal laboratory chemicals to produce novel compounds, which is very useful for the young pharmacist to do the practicals in the laboratories. Thank you very much, sir, for
That's all, sir. See, my aim is, so many pharmacy students are available in your university or other universities also. Some students, they may not get the highly costly chemicals or costly reagents, but whatever the chemicals are there in your university, in your college or other colleges, so with the simple chemicals like a salzlaldehyde is a very benchtop chemical. Benzylamine is a benchtop chemical. Aniline is a benchtop chemical. And AlCl3, acetic acid, all are even BF3 also benchtop chemical. Nowadays, bismuth triplet and BF3 also commonly it is all available chemicals. So with that available chemicals, how best we can make the a novel heterocyclic compounds of the biological interest, so that the student will get some knowledge, background knowledge, so they can go to their laboratory, they can synthesize the compounds. That's what my aim, to give a simple chemistry to some novel heterocyclic compounds.
Yes, sir. Indeed, it is a very excellent presentation, sir. Thank you very much.
If there are any questions, please let them ask. No problem.
Sir, the session is open. Dear participants, the session is open for questionnaire. So you can ask the questions in the chat box, or you can ask directly to the speaker.
I could see that Dr. Gururaj Kulkarni and Dr. Viswaranjan and Vishwanath Mishra, then Tarun Reddy, I could see Mohan Verma. So then Dr. P. Viresh Babu, Dr. Singhal, so Dr. Biswajit Basu, Sarita Reddy, Dr. Mishra. I hope that the session is benefited the faculty as well as the students.
Sir, so currently one of the participants, they're asking, during the research, how can we know that we have synthesized the same compound, and how can we identify it?
Exactly. See, sir, once we did the reaction, for example, you have taken the salzlaldehyde. Sorry, I will tell that one example. You have taken the 2H chromine. Okay, sir. And you have taken the 2H chromine and you reacted with the simple salzlaldehyde. Some Lewis acid catalyzed reaction. Okay. So you got a new spot. You got a new spot. That means you know the 2H chromine where it is coming, salzlaldehyde where it is coming, and you got a new spot. So you have to do the column chromatography to isolate the compound. So you isolated the pure compound. Then you have given to the NMR spectroscopy. Based on the NMR spectroscopy, you can clearly say that my starting material, the 2H chromine, CH2Cl is there, for example. OH is there, and then ester is there, where the signals are coming. That signals are disappeared. Okay, sir. Then salzlaldehyde also signals also disappeared. That means there is no aldehyde, there is no hydroxy, there is no aromatic protons. That means you got a different spectra. So you can take that spectra, proton spectra. Then you can take the C13 also. Then you can take the mass. You write the tentative structure. For example, tentative structure 4H chromine, whether the ring is opened or not opened. You can say no, the ring is open or not open. You know, or simply for example, hydroxy is there, CH2Cl is there. There is a HCl elimination. That means there is a ether formation is there. So we can say that ether is formed. That means you can take the IR. Based on the IR, aldehyd is not there. Based on the IR, ester is not there. Based on the IR, CH2Cl is not there. So that way you can conclude it. And by mass also, you can say that this particular mass is not correlating or matching with the compound, whatever you are written the structure. Then, if you go to a literature, is that any 2H chromines or reactions are there? So you will have some idea. Then what are the heterocyclic compounds were prepared from the 2H chromine or 4H chromine? If no heterocycles are reported so far in the literature, for example, you are lucky. That means you made a fantastic reaction. And you have taken the proton NMR, C13, mass, and HRMS. So you are unable to draw the structure. For example, you are unable to draw the structure. Or whatever the structure you are drawn, it is not matching to your NMR or C13. Then if you can give it to the X-ray crystallography, they will provide the structure. That means you can say that, yeah, it is ring is opened or ring is not opened. It is a novel heterocyclic phenazine or phenoxazine, phenoxylene, or naphthoxazine or naphthoxazole or isoquinoline or isoxazole, whatever it may be, pyrazolone, whatever it may be, you got it. Then it is a fantastic reaction. Then instead of the 2H chromine, you made the 4H chromine, you can test with the 4H chromine also. So that's how you can deduce the structure. Okay, sir?
Yes, sir.
Is it clarified? It is clear? It is clarified?
Yes, sir.
Yeah.
Sir, one more question. Some
Please.
byproduct is obtained.
Yeah.
Byproduct is obtained, and how can we separate such compounds from byproducts?
Yeah. Actually, sir, any reaction is not a 100% reaction. No doubt at all. If any synthetic chemist says that, sir, I got the 100% means no, that's not correct. Any reaction 75 to 85, maximum 90 is okay, sir. Remaining 10% or 15%, definitely instead of the starting material, you will get a byproduct will be getting. Even if you make the drug molecule also, if you make the process also, if you deliver the compound to the industry also, no doubt at all, definitely they will ask what are the byproducts, what are their toxicity. If the toxicity level, for example, 1% or below 1% also, they will not accept, they will not take. First, you should tell that what are the byproducts are forming, what is their toxicity, then only they will take the process, then only they will release the product. Otherwise, they will not. So it is a very important question, how you identify the byproduct, how you know that it is not a toxic compound. Yes, sir. So after doing the reaction, you got the some three spots or 2 spots or 4 spots. Then, for example, if it is a natural product, if you are isolated, you will get the 10 compounds, 20 compounds, 30 compounds is a mess up. But when you do a synthetic compound, you will get minimum 2 or 3 or 4 maximum. So you know what is the major compound. You know what is the major compound. You know what is the minor compound. That means maybe 10% is formed, maybe 2% is formed, maybe 50-50 is formed. Then you are isolated the one compound. The next compound, for example, you got only 50. What about the another 50? Another compound you got the 20. That means you go for the polar, mid-polar, and non-polar. That means first you will get the non-polar. First you will get the non-polar compound. Then you will go to the mid-polar compound. Then you will go to the polar compound. That means one by one, you can separate it. You can separate it very clearly. Then you can take the spectra. Then if you take the spectra, you know what is the compound it will form. You will have some general idea what is the compound. You got the compound. Then byproduct means what exactly it is, what intermediate is forming. That is also byproduct. What I have shown one intermediate in my slides when I am projecting. I have shown the intermediate compound. So that may not be cyclized 100%. That may not be cyclized 100%. Okay, sir. That is only cyclized to 80%. That remaining intermediate is there. You can isolate it and you can get it. Like that, another compound maybe, for example, just now I told hydroxy and chloro. The ether formation is there. That is another compound. So similarly, you can say that one compound I got the 50% yield, another compound I got the 20% yield, another compound I got the 10%. So that way you can make it. Okay, na sir? Hello?
Yes, sir. Sir,
And biological activity also, sir, biological activity also you can test all the compound. And for example, that is very important compound, you can give it to the toxicology study. If it is a toxic, you cannot use. If it is not a toxic, it is showing some activity, definitely you can take it a drug molecule or maybe intermediate that can be used to make another heterocyclic compound. Okay, sir?
yes, sir. Sir, I'm currently working in the field of co-crystals, co-crystal technology.
Yeah.
My question is, uh, so there, I mean, some of the studies which I am reviewing is they're asking for single crystal XRD analysis, which will
Okay.
tell us about the bond lengths in the structure of the compound.
Actually, sir, whenever you take whenever you prepared any compound isolated in a pure form, for example, greater than 90% or greater than 80%, you can go for the crystallization where the impurity will be coming to the solution. The pure crystal will be formed. You are submitted to the X-ray crystallography. They will give entire data, the crystal data, where nitrogen is there, where oxygen is there, where carbon is there, where double bond is there, where ester is there, amine is there, amide is there. Everything they will give, including the whatever the bond angle, bond length also they will give, sir.
Yes, sir. I could not find any laboratory which is providing this sort of support.
No. CSRICT will do, sir. CSRICT will do.
Okay, thank you, sir.
We have the facility, single crystal crystallography. We have the facility. We have the facility, single crystallography as well as the powder diffraction also we have.
Yes, sir.
And we are helping the industry also. Some industry people will come, they will submit the samples to CSRICT. CSRICT is helping them, sir.
Yes, sir. Your inputs will be very helpful for my research work.
Yeah, thank you. No, ultimately, whether it's a professor or scientist, we see that it should be helpful to the society. It should be helpful to the faculty and particularly to student to carry out their research work.
Sir, one question from the participant.
Please,
He's
please.
asking, sir, how we will obtain the product after condensation if they are not precipitating in the final step?
Sir, simply, for example, you have done the reaction and for example, you have taken the solid, okay, two examples I will give, sir. You have taken the liquid compound, starting metal, you have taken the liquid compound, another starting metal, you got the solid compound. It's very clear. It's very clear. By washing also, you can remove these things by pentane or hexane or benzene, something, and you can get the solid. So that way crystallization can be done. Okay, sir. If you put a non-polar compound, definitely these, whatever the substrates are there, that will come to the non-polar solvent. Whatever the compound it is formed, for example, it is a solid that will be solidified and you will get the compound. And that particular compound, whatever solid is coming, that you can give one more wash, you can get the greater than 95%, I can say, pure compound. For example, now I told about the liquid, solid. Then if you take a solid, then if you get the liquid, that is different. If you take the solid, then you can say that, sir, how do I know that all are solids? Okay, all are solids only, right? For example, the reaction went only 60%, 40% solids are remaining in the reaction. All the solid means you have to ultimately go for the column purification only. Where just now I told that one is the non-polar compound, one may be the mid-polar compound, one may be the more polar compound. So whatever the compound you got, that is a more polar compound. Definitely that elutes later. Whatever the non-polar compound is there, that elutes the first one, then mid-polar elutes the next one. Finally, whatever the compound you got it, for example, it is highly polar, then gets the elution finally. So see, you can separate very clearly all the three compounds. No problem, no doubt at all, no problem at all.
Okay, thank you, sir. Participants, sir, any more questions? Ms. Pradeepa replied, thank you, sir.
Yeah, thank you very much. Yeah, sure. You can feel free to ask any questions. I have them. You can give my mail ID also. Whatever the doubts they are having, definitely will be clarified. Will be clarified. Okay, sir.
Okay, thank you, sir. So with this,
Yeah, I am almost. I took 1.5 hours. I think it looks
yes, sir.
like.
We
Thank
started.
you, sir. I took more time. Sorry for that.
No, no problem, sir. It is a very enlightening session. So actually, we got the idea how to utilize the normal laboratory chemicals to produce novel compounds,
Yeah,
because it is a very high time where the research is to be converted into a active research paper or a patent.
exactly, exactly.
That way, it is very important for the country as well as the individual also.
Yeah, particularly it should help the students,
Yes,
student
sir.
community. Yeah.
Basically, everyone will think that we require costly chemicals to do experiments, and
Yeah, for everyone, it is difficult to get the costly chemicals. Sir.
but you have broken the stigma.
It's okay.
Thank you, sir.
Thank
So
you very
I'd
much.
like to
Yeah, thank
propose
you for
vote of
giving the
thanks, sir.
opportunity. Yeah.
I would like to propose vote of thanks, sir.
Thank you.
So good afternoon to all the participants. Uh, I would like to propose vote of thanks to uh, Dr. Chinaraj Garu. Sir, your session has enlightened all the participants about the various chemicals that can be produced with a simple chemical reactions in the laboratory. And you have also given us valuable input on how a basic cross-linkers can be used and how targeting can be achieved. Sir, your session has enlightened us in good manner. So I would like to thank on behalf of the Sri Vishnu College of Pharmacy and Sri Vishnu Educational Society for enlightening our staff, faculty or students, as well as all the participants of the FDP. So thank you very much, sir. Thank you for sparing your valuable time and enlightening the participants. Thank you, sir.
Yeah, thank you very much. My special thanks to Dr. Professor S. Nataraj, your principal. Convey my best wishes to you, to him, and all the faculty. Thank you very much, sir. Now
Thank
I am leaving. Okay, sir. Is okay.
With this one, I am ending the session, sir. Thank you.